Here, what happened if we removed only the leaves? So again, in the stage about 6-7 centimeters before, we removed all the leaves. And then we can see that the growth also is almost normal. Flower buds will be flowering. So in that case, flower buds is responsible for the elongation entire stems. Flower buds is responsible for the stems. So in natural conditions, for the growth of the stems are responsible flower buds and the leaves. Auxin, indolent acid, is produced in the pistil and the leaves. It is transported basipetally and induced the growth of the internode. So in this case, tulips, flower buds, pistils mostly is able to induce the growth of all the stems. We also analyzed the effect of other auxin, artificial auxin. What about their effect on tulip stem growth? Again, we make similar experiments. Here, when the length of shoots was about 6-7 centimeters, all leaves and flower buds removed and no growth more. If we applied 4 and 5 IAA to different concentration, we have normal elongation of the stems. If we applied artificial auxin, but if we applied 2,4-D, 2,4-dichlorophenoxyacetic acid, and indole butyric acid, we have also growth. This last one, it is now, we know that is natural auxin. We can find this kind of auxin in many plants. So it is also interesting that only natural auxin can induce the stem elongation, but also artificial auxin can induce stem elongations. Also, it is interesting is phenomenon of polarity of auxin action. Here, again, similar experiment. We applied, we removed all the leaves and the flower buds, and no growth. But if here I applied lanolin only as a control in the middle of the last internode. In tulip buds, we have 4 internodes, first, second, and third. Highest, we call fourth. No growth, of course. But if I applied IAA in the middle of the 4th internode, you can see that we observe the growth only below treatment. Only below treatment. But no growth in acropolar direction. So it means that only basipetal, which is polar transport auxin, is responsible for the tulip stem elongation. No growth in upper part. Probably we have also found this transport, acropolar direction, it is diffusion, not polar auxin transport, and no growth. If I applied 67 NNA, also we can induce the growth, smaller degree, but no growth above. But if I applied 2,4-D and IBA in the mid of 4 internode, we can observe the growth in both directions, both below and above. Here also, below and above. So it is very interesting phenomenon why the artificial auxin can induce the growth of tulip bulbs in both directions, but natural auxin only one direction. I hope that we discussed with Professor Miyamoto, we will try to little bit solve this problem in near future in our joint cooperation. Sorry, this may be not the best picture, but I would like to say again that here, I would like to say that continuous supply of auxin is necessary for tulip stem growth. Here, auxin was applied when the length, treatment was applied when the length was about 6 centimeters. So we observe the growth different in the dynamics after 1 until 10 days growth of first internode, second, third, and the fourth. In next picture, I will show that the IAA applied here was removed after some days. Here, treatment was made in 0 times, but after 2 days, the auxin was removed from the top of the stems. So after removal, the growth was stopped. No more growth. Even similar results were obtained if I removed the auxin on the third day. Treatment was at 0 times, so again, almost no growth more. So it means that continuous supply of auxin is necessary for this tulip stalk elongation. So we used also IAA in order to labeled with C14. We found that if applied labeled with C14 on the top of the stems, these IAs or metabolites are translocated to the basal plate, and basal and observe the growth, of course, induction, and so it is basal plate. It is main part of the tulips which is responsible for the transfer of the IA or metabolites to other parts. We know in the practice that ethylene, it is very harmful, the harmful effect on tulip growth and development. So here we would try to, in our system, we would like to check what about the effect of ethefone on the tulip growth and development. Ethefone, his name is ethylene chlorophosphoric acid. It is this compound which, after contact with plant, ethylene evolution takes place. So if apply ethefone, we can say about ethylene, because this factor really is ethylene. So we applied here, we removed, when the length of the stems was about, again, 7 centimeters, we removed of the leaves, flower buds was present, and applied ethefone in the first internode, second, third, and under the flower buds. And as you can see, if applied of the second internode or third or anthropods, we have inhibitory effect on stem elongation, and for flower bud blasting, we can observe the result. So really, ethefone, ethylene, it have inhibitory effect. In literature, it is known that probably ethylene inhibit auxin polar transport, not only tulips, but also. We use also another system. Here is 1 and 2. It is control of supply and ethefone on the top. And here IA from number 3 to 8, IA was applied on the top. And here was applied additionally ethefone on the first, second, third, and the fourth internode. So we observed that growth induced by IAA is inhibited by application of ethefone. So against, it is clear here that ethylene inhibit probably polar auxin transport, and growth, total growth of the stems is inhibited. But it is interesting also that we know that we have STS, silver thiosulfate. This compound, it is now, these compounds inhibit the action of ethylene. Silver thiosulfate inhibit the action of ethylene. So here we have this kind of experiment. Control, after removal of flower buds and the leaves. Here we applied IAA to number 3. Number 4, IAA and ethefone on the top of another. So ethefone inhibit the growth. But if applied also additionally here, in the cotton wick was socket wrapped above the stems, a socket silver thiosulfate, which inhibit the action of ethylene, we have normal growth. Ethylene inhibit the growth induced by auxin. We applied additionally silver thiosulfate. The effect was reversed, normal growth. So it is interesting phenomena in the tulips.